Kia ora. Tēnā koutou, tēnā koutou, tēnā koutou katoa. I'm Gary Evans. I'm the Director of the Ferrier Research Institute. And look, thank you so much for this turnout. We really appreciate the support of both this lecture series in honour of Robin Ferrier, the Institute itself, the University, Te Herenga Waka, Victoria University of Wellington, and of course the science system in general. We're going through interesting times. I have a limited number of tasks today. I am going to be your MC. I will, at the end, help with questions and answers. I won't provide the answers, but I will definitely relay the questions. But my most important job is to invite Vice-Chancellor Nick Smith, Professor Nick Smith, to come up and introduce our Ferrier lecture for 2025. Nick's been in the role now for a couple of years, arrived at an interesting time for the university. He's done a fantastic job, been a brilliant leader, has really looked after the research institutes themselves. So I just want to say publicly, Nick, thank you so much. But I would ask you now to come up and introduce this year's Ferrier lecturer.
No mai, haere mai, piki mai, tēnā koutou, tēnā koutou, tēnā koutou katoa. So, as Gary said, it's my absolute privilege to be the Vice-Chancellor here at Te Herenga Waka, Victoria University of Wellington. And tonight it's my particular privilege to welcome you all to the 12th Annual Ferrier Public Lecture. I'd like to, in addition to welcoming you, acknowledge Duncan Ferrier, the son of Robin, who has joined us tonight, has taken some time off from his law career to learn some carbohydrate chemistry, who, along with me, will be, I'm sure, watching and listening carefully. This is a prestigious lecture series, which, as many people here will be aware, is named after and named in honour of Professor Robin Ferrier, who was a world-class chemist and an esteemed and much-loved member of our university community. He played an important role not only in research and in teaching, but in the kind of things that university academics do when they transcend their field of actually producing the next generation of chemists here in Aotearoa, New Zealand. This public lecture was established to recognise Robin's significant contributions to both chemistry, both here in Aotearoa and internationally. And I think it also serves as a heartfelt tribute to his memory. From his university, from his friends, and from his colleagues, and I'm sure many people here who knew him personally. This evening's lecture is titled The Sweet, the Sour and the Spicy Sides of Microbial Glycans, and will be given by Professor Cristina de Castro. Professor de Castro first completed her master's degree in 1992 and then her PhD in 96 in chemistry at the University of Napoli in Italy, followed by postdoctoral research at the Carlsberg Research Centre on carbohydrate structures in both beer and yeast. She's now a full professor in organic chemistry, and her research focuses on carbohydrate structural chemistry, with over 170 publications associated with many international collaborations around the globe. In addition to presenting the Ferrier Public Lecture, Professor de Castro is spending time here at Te Herenga Waka, engaging with staff and postgraduate students on our research. Thank you for being here. We're delighted to have you here. Professor de Castro said she was also looking forward to seeing our country because of its connection with Lord of the Rings, and so I hope you get to see some magic and also some scenery as part of our beautiful place. Professor de Castro's lecture, as I've said, will highlight the relevance of carbohydrate structure in different areas and cover a range of what will be, I think, really interesting examples. Sweet glycans that can exist in bacteria from the gut, sour glycans that can enable pathogens to elude immune surveillance, and spicy glycans, where discovery has triggered many open points in glycobiology. Lastly, I'd like to express my gratitude for tonight's sponsors and for everyone who's made this event possible. The Ferrier Lecture is funded by an endowment from Dr. Pepe Prassett, a former PhD student of Robin Ferrier, and someone who I was delighted to have lunch with just at the end of last year, and he talked fondly and passionately about his time, not just here in Wellington, but doing his PhD with Robin. He himself has enjoyed a distinguished career as a biomedical scientist and entrepreneur, and has established this endowment to honour the world-class education he received in carbohydrate chemistry and his research under Robin's mentorship. It is now my pleasure to welcome Professor Cristina de Castro to the lectern to deliver the 2025 Ferrier Public Lecture.
So it's a big honor to be with you today, and I'm going to tell you some things about sugars. So, as you see, the title of my talk was quite peculiar, and I have organized it in the way that I will give you a brief introduction on carbohydrates and the small secret you need to know about the structure and the way to do it. And then we will turn into the sweet side of carbohydrates by looking into gram-negative bacteria. Then the sour side is not very pleasant, and that's on viruses. And we will see also some special viruses for the spicy side at the very end. It's good to have some spices to wake up. So, about the introduction, and please note here there is the structure of two different monosaccharides, so glucose and galactose. And for a trained eyes, like a chemist can be, it's easy to see that there is one tiny difference between them. So it's just the orientation of this chemical bond. So these monosaccharides are the building block of polysaccharides. And so look that the tiny differences in the end there will be big differences in terms of biological activities and properties. So imagine for chemists to get to deal with them when the differences apparently are so small. So things have been simplified, so you don't have to, have this structure through all my talk, because now it's possible to make use of a simplified representation. That is the one developed and known as a symbolic nomenclature of glycans. In this way, it's much easier to identify glucose from galactose, simply because we have symbols and colors that are very different for the two types of monosaccharides. And so that's a good way to have structures drawn on a paper in a way that is compact, and you pick up differences and also analogies in a simple way. So maybe some of you maybe are thinking in this moment that we have too many symbols, different in colors and size and shape. Well, if you enter in the world of microbial glycans, then there is a very extended repertoire of sugars, and all these symbols are not enough. So they don't cover the monomer space, as we can say, in carbohydrate, in bacteria. But luckily for you, you won't see many, many symbols, so we don't, exit from those that you see here. So what about the structure? Because there is a pipeline that has to be respected for a structural chemist. Of course, it starts with the isolation of the material and the purification from the source. Then, clearly, we need to understand which kind of monosaccharides are present in the sample. Also, the way they are linked. That's important. But then we reach the end with the nuclear magnetic resonance, the NMR analysis. That's key to understand the sequence between the different building blocks and other parameters that are key for the kind of polysaccharide, like the size of the monosaccharide, and the way it is configured at the magnetic center. So these are technical details, but they really contribute to define the structure of a polysaccharide. And once you know the structure, you can move further with deeper questions. That's quite interesting, because then you can ask to the system, what is the biosynthetic path? So you can start to learn about the activities of the glycosyl transferase that are involved. So you enter from this pipeline in what are the biochemical studies, and then you can make also association of structure to function to the enzymes that are involved. Next, what else you can do? You can understand something about the role and activity of these glycans. So do they have any good activity, so a pro or anti-inflammatory activity? That is the not good and the good one. Or also which proteins they are interacting with, and if there is any signaling cascade downstream. Other things that can be determined, like you can run into exploitation of polysaccharide. Many of them are really powerful molecules as a postbiotic. So you take them, that's good for your gut, basically. So they can be exploited. And the same holds true for vaccines. Many vaccines are based on carbohydrates, so you need to know what you have in your sample to use it. And now there is the new frontier in material. And during my talk, I will give you some example in role and activity. So we will stay in this area with some structure coming along with it. So let's start from the gut. You know, because now everything seems to be gut related. I don't know if it's also you have this feeling. But if we look inside ourselves, where they say that there is our second brain, basically, our stomach, so we find a lot of microorganisms. There are viruses, or to be precise, there are phages, so the viruses of bacteria. There are archaea, some few, and also some fungi. And sorry if I don't know if I never know if it's fungi or fungi, so apologize me for that. And then we have a lot of bacteria. Many, 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 really, you can see that. And so this list is only considering bacteria, different types, that can be more or less divided into gram-positive, those that you see in the blue circle, and in gram-negative, that are this other. So the name gram is not an acronym, I want to be clear. That's the name of a Danish microbiologist who developed a coloring procedure of the bacteria. And so that's why the name gram-positive and gram-negative. The gram-positive bacteria, so from gram, the microbiologists, are able to retain a certain color, the crystal violet. The gram-negative, they cannot. And that's depend on the way the bacterial membrane is constructed. So you can see there is a different architecture. I don't want to enter into the details of this architecture. It's just clear that they are different. But what is important for us is what stands on the top of the membrane. In both cases, because you have here these sort of chains, these are carbohydrates. And you can find them both in gram-negative and in gram-positive, so they are densely covered of glycans. That is important to know the structure at some point, of course. So, what next? Let's start to have an idea of what are these molecules and also the way they are classified. And in this regards, I prepared, these slides. And these are the main carbohydrate molecules that we can find. And some are present only in gram-positive bacteria, like the wall-tecoic acid and lipotecoic acid. For us in this moment are less interesting. So you see, I also did not use any color. So they are not good. But basically, the main structure is a polyol phosphate. So there is a chain of carbon atoms and phosphates as a diester, and some monodisaccharides in the typical pattern. Then we can find the isopolysaccharide and capsular polysaccharide both in gram-positive and in gram-negative bacteria. And the difference between them is that when we have a capsular polysaccharide, it's really linked very strongly on the bacterial surface. And for isopolysaccharide, the opposite is true because they stay on the surface very loosely, so not very well, and so they are lost in the growth medium. They are released, basically. While the last molecule, this LPS, so it stands for lipopolysaccharide, is unique in gram-negative bacteria. So again, we have a polysaccharide part that is drawn here, that is very long, so we have a repeating unit. In the same way, we find it in capsular and isopolysaccharide, repeated many times and making really the molecular weight of the molecule. Then we have a small region that I see, as you can see, I put a little bit on the side, that is called core of the molecule. And finally, there is a glycolipid. So a domain, I would call it, in which we have two sugar, the phosphates, and the lipid. And this one is inserted into the bacterial membrane. So why I'm giving you so many details? Because there is one thing to consider. If we take all the polysaccharide parts that you can see on this side and now circle in a box, so this structure can be really very heterogeneous. So, and that's why I use a wealth of colors and symbols to denote that we have many different sugars that are making them. I mean, these are structure just pure fantasies, just to give you the concept. So there is high structural diversity and also very low level of conservation. It means that we take two bacteria in the same species, like Escherichia coli, but different strains. So it's like two persons of the same place. Then they will have lipopolysaccharides that are different in this region, or capsular polysaccharides that are totally different, although they are genetically closely related. And that's true for all polysaccharides, so they are not conserved at all. So what changes here in the core region? Then we find a certain level of conservation. So you see how this core region is a little bit outside. That means if you take these two different E. coli strains, genetically very close to each other, the core region will be very similar, if not identical. And if we move a little bit further and we look into the lipidae, so this glycolipid, then the level of conservation is very, very, very high. Truly high, so we take Escherichia coli and we take Bacteroides, for instance, so they are genetically very, very distant, but the lipidae will be very similar in all of them. And that's why for this molecule, we use also for this moiety of the molecule, we use the acronym of PAMP. That's an acronym that is used for class of molecules. What is a PAMP? So, PAMP by itself means pathogen-associated molecular pattern. And this acronym has been taken different shades and also some variation through years. But the first one that was invented was this, and I will stick on that. So what is a PAMP? What is this associated molecular pattern? So by definition, it is something that is very conserved among different species, as I told you before in the lipidae. So it's also something that is essential for the survival of the organism. So it cannot simply mutate it, because if it mutated, maybe it's not functional anymore, and that's lethal for the organism. It won't be viable anymore. It will not be able to live, to replicate. It will be its end. And that's why a pump, any type of pump, is almost not susceptible to mutation. There can be tiny, tiny variation, but that's it. So the general motive will be that. And together with the concept of pump, we have another that you will hear quite often today, afterwards, and that's the PRR. Again, an acronym, that's pathogen-related receptor. Because for any pump, here, conserved domain, there will be a receptor on our cell that will recognize it. So these receptors are very, very important because they are part of our innate immune system. So they are active since the very first day we are born. That's our first line of defense, and it's going to protect us against many threats. How many there are? A lot. And during this talk, we will meet the toll-like receptor, the TLR, plural, there is more than one, many, and also the DC sign receptor. And if sometimes I say DC sign, it's because I'm Italian, and so G, that's G, not C. In my language, I will try to avoid it. So our pump, the lipidae, I show you the chemical structure, sorry, but it's just to show that we have two sugars. These are glucosamine, they're very similar, and there is what we call the polar head on one side and the other that are the phosphate units. And relevant for us, there are also some fatty acids, so compounds with many carbon atoms, a certain number of carbon atoms, and a number you can find it here in this circle. So in E. coli, what is distinctive is to have a length of 14 carbon atoms, except in one case, in this position. And this lipidae is recognized by the TLR4 receptor, the number 4. There are many others, but that's the one for lipidase, until a few years ago when things start to change it. So TLR4 recognized E. coli. Well, actually, we have the formation of a quite peculiar complex because there is a dimerization. And all these triggers signaling to the nucleus and production of other molecules. Some are good, some are not very good. And this is the case of the inflammatory cytokines. And that's a point when you hear about septic shock, no? Then it is an uncontrolled release of cytokines that can be induced from lipidae of bacteria that are circulating in your body in that moment. So this is something that absolutely we don't like. So it's we are very much on the risk and on the sour side of molecules. So at this point, a question for you, maybe just to reflect a little bit. So is the lipidae only a single molecule? in the sense that I take E. coli, it's producing the lipid is that one molecule only is making a complex blend of lipidase. Let's put a plural at this point. So the answer to this first question is that there is not only one lipid. And this is clearly visible by this experiment. This is a multi-spectrum, so basically it's a mass spectrometry. Each line has a number associated. That number is the molecular weight of the compound. And the molecular weight depends on its structure. And you can see that we have many different lipidase. And they differ for the number of lipids. Here we have the classical. You see these 6 lines means that we have 6 fatty acids. The red dots are the phosphate, and there are the 2 sugars. But there is also a species where the phosphate is missing and another in which we don't have the phosphate. And you can count a lesser number of fatty acids. So what we have in the end is a family of molecules. Each bacterium, when produces the lipid, makes a family of them with tiny, tiny differences. And so the next question is, do you think that all these different molecules that are all together making the membrane, they have the same activity in terms of inflammation ability? No, they don't. If this was the things that you were imagining, you were right. So, and this has been proved. As you can see, this is giving more or less an idea of what's going on, because we have our lipid with 6 fatty acids, so the classical one, let's say, that is recognized by the TLR4 receptor, and it's triggering 2 different signaling pathways. Note the color. One will give you inflammation, and the other one that is activated at the same time, so both together, will give you the immune activation. That is something good, because this immune activation at the very end will go into the adaptive immunity. So we can give you the production of antibodies. So that's a good thing. And for this lipid in which we are missing the phosphate on one side, then we have only one signaling pathway, so the good one. We start to like this thing at this point. And also changing the number of lipids. There are these species that are particularly, they take the name of lipid 4A. So they can be recognized, but they give no effect. I'll tell you, this kind of lipid is produced by Yersinia pestis. It's not a very good thing. So all this to show you that the activity can be tuned by structure. And we have to keep this in mind all the times we have a molecule in front of us. So in this case, we have that it changes depending on the number of lipids and the phosphates, the number of phosphate and so on. So definitely we have that structure makes the balance between, well, the difference between beneficial, our sweet, and harmful. Okay, this was a long premise. But just to make you think that we have this lipid from gram-negative bacteria, and we are colonized by many gram-negative bacteria, but none of us suffer from inflammation normally, I mean, if we stay well at least. And in this slide, I showed you which is the population of gram-negative bacteria in our gut. There are many. And in the yellow one, we have those in the phylum of Bacteroidales, and there are a lot, with the Bacteroides vulgatus as one of the most abundant, because I'm going to return on this bacterium. Then we have Protobacteria, where is E. coli, Escherichia coli. So the one is giving inflammation. There is also some representative in Verumicrobia, or Verumicrobiota, as they were changing the name recently. We have Akkermansia municiphala. So we have all these gram-negative bacteria with lipidase all together, but we are okay. It means that something is going on there, and maybe the combination of all of them is such that we don't suffer by inflammation. So basically the question is, what about the effects of all these lipidae, of these molecules? Maybe there are some counterbalances. And an answer to this question came a few years ago when the lipidae, well, actually the lipopolysaccharides, all from Bacteroides related species, were compared to E. coli, Escherichia coli. And I show you here the structure. This is the one from E. coli that I showed you already many times, and this is the structure of the lipidae from different Bacteroidales. You can note there are some analogies, of course, but also differences, and these are key. So first of all, there are different types of fatty acids. I told you, the molecules are very similar. We have very tiny variation. Tiny variation can be big differences at the very end. So in E. coli, we have fatty acids that are 14 carbon atoms. Instead, the length here is higher. So we have 15, 17 carbon atoms. And please note that there are, these small things at the very end. These are branches. So it means that it's not a linear, but at some point there is like ramification. So other structural difference, small but relevant. And the number, that's another point. The inflammatory lipid A has 6 fatty acids. Here you cannot count 6. We have 4, we have 5, but we don't have 6. And also the phosphates, the polar heads, that we have 2 in E. coli. Then the number is lesser in this other case, and there is 1 missing most of the time. So what happens with activities? And the first readout is the inflammatory properties, and the blue line is the effect given by the lipopolysaccharide, so the lipid A also from E. coli. And here you can note that we have all a cell line that is much higher compared to all the others. And all the others instead are the lipopolysaccharide from the Bacteroidales. So you can see how E. coli is given inflammation, the others are doing nothing. That's a good news. So at least we have only one bacterium, one species that is giving inflammation. So it's a minority. We can be happy. No, we can be more happy if we look into the next experiment. That is a competition experiment. Because what has been done? So to the system, the lipopolysaccharide from both the Bacteroidales, they have funny names, yes, I agree, and the E. coli were given together. So there is the control in which there is only E. coli, and you see this level of inflammation. This is the reader, let's say, readout. But when you start to mix E. coli lipopolysaccharide with the lipopolysaccharide from Bacteroidales, at a certain point you see that the level of inflammation decreases. It means that these bacteria, these good bacteria, have lipopolysaccharide that are neutralizing the inflammatory activity of E. coli lipid A. And that's why we are very happy and we don't suffer from inflammation. So these are good bacteria, really, for many, many reasons. And so far I've been talking to you only about the lipid A, but the lipopolysaccharide contains also something else. So now we enter more into the detail and we look in the specific one of these Bacteroidales, and we take Bacteroides vulgatus, that is one of the most representative, was at the higher bar. And you see here, everything in the LPS counts. So like many things, you don't throw anything away because everything is useful. So what happens? So first, the full structure of this lipopolysaccharide was determined by using a combination of MALDI, to remember the one with lines and molecular weight, and also nuclear magnetic resonance to establish the sequence between the glycans. Great. So we have a quite complex structure. Just to comment a little bit, because it will be with us for the next 5-10 minutes, more or less. And so what we have a lipid A on this side. So the 2-glucosamine, the fatty acid, like we saw before, reduced level of phosphorylation. And you see here also the pictorial form that maybe is nicer to be seen. Then there is the core region on this side with strange sugars like this KDO. KDO is a sugar with 8 carbon atoms, so it's quite big. And it's also considered the hallmark of gram-negative bacteria because it's there and it's part of the lipopolysaccharide. And this was a very strong belief until a few years ago when it was discovered in plant. You have to change mind at some point in science. But anyway, KDO is here, there are other sugars, and this is the O antigen, the true polysaccharide. And for simplicity, it is represented as a repeating unit that, of course, is assembled in several copies. So to study the activity of this lipopolysaccharide, what has been done has been also to dissect, so to split it in parts. so that it was possible to see the full molecule and also the separate component, and at some point also some representative structure that were synthesized chemically. So it's important to have all the things together into work. And to split this molecule into its part, it's very easy because the cation linkage that is here is very labile. So you need just acidic condition under controlled temperature for a certain time, and then you split the lipid A part from all the rest. So the core region plus the O antigen. So this can be assayed separately, and this was what was done on a suitable set of cells. And the activity versus the TLR4 receptor, as you can see, for the lipopolysaccharide as a whole, or only the lipid B moiety, basically was zero. There was no activity at all. While, as you can note, the lipopolysaccharide for E. coli is active at all doses. But then the interesting finding was that this lipopolysaccharide was active against another type of TLR receptor, the number 2, which was considered instead selective for lipoproteins, so other type of compounds. Yet, this lipopolysaccharide as a full molecule is active against it. If we take the lipid A, that is this line here, no activity at all. So we need a full molecule to observe something. And another point is that if the system contains both the TLR receptor, the 2 and the 4, then there is a sort of synergistic effect. So we observe higher response if there are both the TLR receptors. So this molecule is quite peculiar. It means they can interact with more things, and also maybe to bring them together to get a sort of synergy. And last point, important, of course, going into competition studies, the one that I showed you before, the whole LPS is an agonist, so it's able to reduce the inflammation, confirming the other data that were seen more in a different way, in a different cell system. So what next? Because this idea that there is interaction between lipopolysaccharide and different receptors, maybe all the things are put together and they interact in, multi-complex, drove the question about the involvement of other PRR, other pathogen-related receptor. And then the interest fell on DCSINE. DCSINE is able to recognize carbohydrates. So he has a lectin domain where there is a calcium ion that is important for recognition. And also it has been described that mannose and fucose are the sugars that are better recognized from this receptor. And actually both fucose, you see here this red triangle, and mannose are part of this molecule, of this lipopolysaccharide. So the question is, once again, always question, well, otherwise science doesn't move on. The question is, what is recognized by this lectin? The antigen, the core region, both. Yes. Say both, you will never be wrong. So it's possible to see in this ELISA test that recognition occurs with the LPS as a whole, very well, very intensely actually, with the antigen, the core, and the other dots here are just synthetic compounds that were produced to see specific effects. So what happens? The recognition. And so to this end, to understand the role of the O antigen, it was possible that this oligosaccharide was synthesized and assayed in a DCSINE system. So we have an oligosaccharide that is simple. It contains mannose and rhamnose that are in the repeating unit of the O antigen. And it's also short because it was, after it was discovered, it was not really important to have a long oligosaccharide. And to understand if this oligosaccharide was recognized by the receptor, by DCSINE, it was possible to perform this kind of measurement. So it was determined by going through this kind of measurement that is called saturation transfer different NMR, so nuclear magnetic resonance. So this is the output that you observe, basically. And on the lower part, there is the normal NMR spectrum that is depicting more or less the proton that are in these molecules. So we have peaks in different regions, and each one is associated to a proton of a sugar. This is just to give you some basic information about it. When we look into the spectrum with interaction with the protein, you can note that some of the peaks are still there, and some instead has totally disappeared. Why this? The peaks that are still there are related to protons that are interacted with our lectin, the DC sign. If they disappear, it is because that part of the molecule is not interacting with the receptor. That's the way to read this experimental data. And maybe to a careful eye, it's possible to note that the intensity of this peak is not always the same. Some are very intense, some are less intense, and some are really fading in the baseline. So we can have a criterion to classify this peak like a strong intensity, medium intensity, low intensity, and we can use colors to do that. So if they are very intense, like this catalog as M4, we can associate the red color, otherwise we use orange, yellow, none, if they disappear totally. And we can match these colors on the structure so that we see in a pictorial form what is really involved in the binding with the DC sign that is here is sort of Pac-Man. But the criterion to have this drawing is that this is a binding cavity that is complementary to this part of my molecule. So the red part is what is closer to the cavity, to the binding pocket, also as a term. And all the rest you see has no color associated, so it's not binding to the lectin, it's very far outside, and that's the way it is. So my DC sign likes mannose when it is in a terminal location. And what about this other oligosaccharide? So if we scramble it, so we don't have mannose anymore on the terminal side here, then there is no interaction. So DC sign need definitely for the recognition a mannose unit that is here linked to nobody else than a rhamnose in this position. And if it is moved on the penultimate or any other, then the DC sign doesn't like it. So with this same approach, it was possible to determine also the binding with the core region. And once again, here it is used a synthetic molecule because synthesis is important since in this part, on the terminal part, there is a rhamnose and not mannose. So the effect seen, it's only that due from the core region. Once again, there is the STD-NMR spectrum. So, and you can note, compared to the reference spectrum, that there are many, many signals that appear really intensely. So it means that also the core region is recognized by the DC sign receptor. I remember you, the DC sign receptor is a receptor of the innate immunity, so it's contributing to our defense. So what happens using our color code? You can note that there are protons that are red, others that are orange. So some sugars are close to the binding pocket of these lectins. Some others are less close, some are totally outside. And between the sugars that are making a very strong interaction, we have fucose that is here. This is a 3D model obtained by molecular modeling. And fucose is engaged in interaction with the calcium ion that is this ball that you see here. That is bridging on the other side with the protein itself. Then we have on the side the galactose in the furanose form and another in the pyranose form here, and KDO also is having contact. So as this part of the study, what is seen is definitely all the interaction I mentioned you with this monosaccharide. But what is really interesting is that the lipid A, so the toxic part, well, the one that was interacting with the TLR4 receptor, is located here on the KDO residue. And when it is mapped over the protein surface, it just stands outside and it doesn't go into the binding pocket. So it's there outside and free to interact with any toll-like receptor that is around. So this is the full view of this LPS. Everything is doing something. So we have a dual-sided molecule because the DC sign can link this end and also the other end. So we can have the coordination of two, actually. So another point that is important, that we have the lipid A moiety that remain exposed. So it can interact with the TLR4 or the TLR2. We don't know that. It has to be determined yet, new avenues. And the relevant fact for us is that from this side, it's possible that the TLR will arrive. So it will start to cluster with the DC sign. And that's an important thing in biology always, because when you have clustering of receptor, this induces a modulation in the response. So it can turn maybe a good response in a very good response, or maybe you can turn it down. So this is something really open field into the understanding of what the lipopolysaccharide and carbohydrate structure can do, and also their beneficial role can so be put on molecular basis. At this point, I think that I gave you enough reason to think that we have sweet polysaccharides. Actually, I showed you only two structures, and also harmful glycan derivatives. Only showed you one, because the situation, of course, is very much richer than I showed you, but there is no time to see everything. And so I just show you here this capsular polysaccharide that comes from gut bacteria, lactobacillus. So, I mean, we also take ferments, nutraceutical, based on that with our probiotic or bifidol. So they all exert beneficial action. This has been proved in both my system and other assays. Instead, regarding the harmful, the sour, there are other motifs that are really dangerous for us, like this alpha-gal, or also this, I show here as a monosaccharide, that is a monosaccharide related to sialic acid. There is the substitution on this amino function given by a glycolide residue. So all these motifs, apart from the lipid A that we don't have, are all motifs that we don't produce, that we see as a foreigner structure. So they are said to be non-self. And so far, all the examples I've been doing to you have been regarding non-self structure that we perceive in a way, sometimes in a sweet way and sometimes in a sour way. So then what happens about structure that are considered as self? So self-structure for us are the sugar that we produce normally during our biochemical processes. So since we do them, we don't consider them as a danger. Of course, otherwise there would be an autoimmune reaction. But unfortunately, there are pathogens that exploit this. So they exploit what we call the mimicking strategy. So they use glycans that are very similar to ours, so that our immune surveillance doesn't start at all, because they think that they are not enemies, but unfortunately they are. And this happened with a certain number of organisms and entities. And for sure, who are really professional mimickers are the viruses. And I show you in this picture, well, this is a SARS-CoV-2, but this is valid for basically for all human viruses, human pathogens. So there is the entry of the virus in the cell. In the case of SARS-CoV-2, it occurs because there is the spike protein that is engaged in interaction with the ACE2 receptor on our cells. So there is the endocytosis, and then the infection process can start. With the infection process, so the virus that is, the perfect opportunist, so it uses the host, is, I don't have the word in this moment, but it's exploiting our machineries, is a parasite. That was the word that I was forgetting. So it's a parasite, and so it's exploiting our resources to multiply. And then after, when it has finished, it will exit our cell, and maybe we'll feel sick about that. So when it's exploiting our resources, it's exploiting really all resources, including the glycosylation machinery. That means that this virus, like all other pathogens, at some point will have proteins that are covered by sugars that are like our sugars, like our oligosaccharides. And so they will looking at least at the end glycosylation of protein, we will find the high mannose oligosaccharides that are these, the complex oligosaccharides in which there is also sialic acid, and also the hybrid that have the same characteristic of the two. So they will look afterwards like something similar to us. So that's the mimicking strategy. And SARS-CoV-2 is even smarter because it's used this sugar in a functional way. So they have a functional role important for its infectivity. So if we look a little bit deeper, SARS-CoV-2, that's the shape of the shape of the virus. And we can see this blue thing on the top that are trimer of the spike proteins, also termed S protein, clearly covered of host-like glycan. And there is one in particular on the asparagine 234 that is key for the activity of, or for the infectivity of this virus. If we look a little bit more in detail, the spike protein, we have that it is covered by sugar, and they are these blue clouds, while in cyan we have the polypeptide backbone that you can see here in the naked form, virtual. But you can note that there is this small part that takes the name of receptor binding domain. This is key for the interaction with the ACE2 protein. And for this interaction to happen, the receptor binding protein has to be in this up position. So it has to show up, literally. And this form is stabilized by the glycan, this oligomannose on the asparagine 234. Why? This was proved by extensive molecular modeling studies. So it took a lot of computational effort. And what is the right perspective to see the things is this image of the model of the spike protein, the trimer, and there is this small cavity here. This small cavity is key. Is located nearby, trust me, this is the nitrogen 234, if I see well, yes, at this distance. And here there is my oligomannose that during the simulation start to move, and at a certain point it's like crawling from this position, from outside, pointing outside to the position of the cavity. So it's going to feel it. And when it feels it, it just blocks the receptor binding domain in the up form. So the spike protein is able to engage the interaction with the ACE2 receptor and to carry on the infection process. This has been proved by deleting, making the mutant protein and deleting this asparagine so that it could not be glycosylated anymore. And so in this mutant protein, the receptor binding domain was down, so was not able to engage any interaction. And that's very sour, unfortunately. Our glycans that are exploited by pathogens to do what they want, not what we want, unfortunately. So, viruses. Viruses are small entities. I told you a lot about SARS-CoV-2. I can tell you even a lot about HIV. And what is common to these two viruses? They are both single-strand RNA viruses. So they are small, and they have a genome that is about some kilobases, 10 in the case of HIV, 30 in the case of SARS-CoV-2, a little bit bigger. And for your information, the size of the genome for SARS-CoV-2 is one of the biggest found so far. So it seems that it's sort of limiting. You cannot have bigger genomes for RNA viruses. So what else? As I told you, these capsids are glycosylated by the host machinery, so they look very much like us. And HIV, despite it has a smaller genome, it's even more extensively glycosylated if compared to SARS-CoV-2. Anyway, viruses are not all the same. They have very different features. They are somehow also fascinating entities. I don't dare to talk them organisms because they are not alive. And I can show you here others. So you can see this is a chlorovirus. The name is PBCV1, where the acronym stands for Paramecium bursata, that is a protist. Chlorella, this is a green algae. And V, at the very end, is virus. So this one is different, at least in shape. This is icosahedral, while we have these things that are protruding outward. And PBCV1 is bald, in the same way like CROV. I can speak also about that, giving you information later. And but we have also other shapes. For instance, these other viruses, the don't move. These other viruses have an icosahedral capsid as well, but they are not like this because these are bold, and these other instead they have a sort of hairs. So they have what are called fibrils. And the fibrils can be spare like this, or very thick and long. And we can also have viruses with tails, like 2-1 virus. And this is the 3D reconstruction of the 2-1 viruses. So you really have an idea about what these hairs are. They're not what they're doing, but they exist at least. And the shape doesn't stop here. We can have different things also. And you can note that we don't have any more an icosahedral. We have something that is more elongated, bacterial-like. And that's the case of pitovirus and cedrat. These two are very much genetically related. And you have a sort of cork opening on one side in pito and two in cedrat. Pandora. Pandora is a fantastic name because if you think to mythology, Pandora, the Pandora amphora, actually was disclosed in many, many surprises, maybe not all good or interesting. And that's why this virus took the name of Pandora, because when it was discovered and studied, it gave a lot of surprises. But it is not a human pathogen, so don't worry. So, and after you see which surprises. And then we have mollivirus, like a balloon shape. And the second name of mollivirus and pitovirus is Sibericum. And because they both were found in ice in Siberia, and this ice was 30,000 years old. And after all this time, these two viruses could replicate, so infect a host. And so that was it. But luckily also they infect Acanthamoeba. So who cares? It's a protist. They are not human pathogens anyway. So one question to you. Did you notice that these viruses are bigger and bigger and bigger? So it was not intentional, really. I didn't draw them bigger because I want to hit your attention. They are bigger. They are all drawn at the same scale. All of them. So it means that Pandora is much bigger compared to SARS-CoV-2 or HIV. So the word that it comes into our mind, looking together and comparing the sizes, that these are giant, because so big compared to SARS-CoV-2. And actually, this is the correct word. These are giant viruses. So maybe some are less giant than others, but compared to SARS-CoV-2, HIV, they're much bigger, and it's not only the physical size that counts. So, but if we look into the genome, we find more surprises. So the genome of these viruses is constituted by double-stranded DNA, the same kind of genetic material we have, basically. And if we look into the size, the smallest of them has a size of 300 kilobases. So it's 10 times more complex compared to the SARS-CoV-2. Surprise, surprise. Pandora is the biggest of all of them. That's why the big surprise, because we have millions of bases. And then, you start to think, okay, what are doing all these viruses with this big DNA material? How they are using it? Well, that's the big question, because many of the genes that are encoded just match with hypothetical proteins. So they are protein, but nobody knows what they are doing, because looking at homology in the databases, there is not much, basically. But for those where the function can be given by annotation, it's possible to find a lot of glycosyl transferases and also other genes that are able to manipulate sugar at different levels. So for these viruses, and differently from HIV and SARS-CoV-2, now there is the belief that they have an almost autonomous glycosylation machinery. And note that I put almost in brackets because, they are still viruses, so they are parasites, they need a host for some resources. But they are able to synthesize sugar, manipulate them, and assemble them together. And for few of them where the study has been carried on, the glycans are different from those of the host just because there is a different machinery to make them. And I'm going to give you very short information on PBCV1, the smallest of them, still with many surprises. So for PBCV1, the current knowledge is that there is a capsid highly glycosylated. You see the structure here in the pictorial form, it's much nicer. So we have a complex oligosaccharide that consists of 4 glycoforms. It means that there is some heterogeneity. And that stems from the fact that this monosaccharide that is an arabinose and this other that is a mannose are there as non-stoichiometric units. It means that sometimes they're there, sometimes they are not in some species. So there is a variability. We have also a highly branched structure with, you see, high variety. These rhamnose units, they have opposite configuration. So they are not the same, in few words. And if we look into biosynthesis, then the enzyme is encoding, the virus is encoding all these enzymes. And these enzymes are used to assemble these glycosidyl linkages. So it's autonomous. And if we look a little bit further, we find this asparagine that is located into this sequence. So the major capsid protein is actually glycosylated at 4 different positions with this oligosaccharide on this asparagine, in which you see the other amino acid that follows. And last, if we take different chloroviruses, then they are all glycosylated and the glycans are different from this one, while the sequences are similar. Okay, so these are the premises. And to understand why these premises are important and why, so I told them to you, basically, we have to look things in a wider way. So I'm summarizing here the information for PBCB1, the structure, and the glycosylated sequence. And I want to turn your the attention on the life domains. So I took this very nice and colored tree of life because it was good to catch attention, basically. And what happens in life? We have here the bacteria, the archaea, lower level of evolution. And then we start to climb this tree, and then we have mammals, men, humans at some point. So what about sugars in all life forms? Well, if we look into sugars, all life forms are glycosylated and they all present glycans. I remind you that viruses are not alive anyway. So in life domains, there are proteins that are unglycosylated. And if we look into the way these are synthesized, the mechanism can be really diverse. So they change from bacteria to archaea, and they change, of course, if we go to eukaryotes. So we pass from simple enzyme to complex with many enzymes all together to determine the specificity. But there is one thing that is the same in all life forms, and that's the unglycosylated sequence. So that's the same in all life form. And there we have asparagine, followed by an amino acid. Then it can be everything is X except a proline. And afterwards, we find or a serine or a threonine. That's it. We don't escape from this situation in all life forms. Then, if you really move your eyes in this direction, then you see the sequence of the virus. They are totally different. Okay, it's not alive. But then why is, first of all, having a glycosylation machinery? Then why this glycosylation machinery is installing with oligosaccharide in a typical sequence? And so the question is also from where the chloroviruses took this glycosylation machinery. We start to have deep evolutionary questions because we don't know where these viruses are matching on the tree of life. So it's close to archaea, close to bacteria. So these are real questions that are important to understand evolution of life in general. We have a trait that is common to all forms of life, that is N-glycosylation, that here is just made in a different way, because looking into the genome of chloroviruses, there is nothing that matches the glycosylation in any form of life. So all these are curiosity-driven questions, of course, but these kind of questions are those that make really spice the life in carbohydrate science. And with this, so I close my lecture. And I'm glad to give you a brief introduction about the group where I work to, that is made by 5 people as permanent staff. And today you heard about the work of Alba Silibo and Flaviana Di Lorenzo for Bacteroides vulgatus. Some of the structure I showed you about the gram-positive bacteria, important and good for our guts, come from the collaboration between me, Professor Molinaro, and other scientists. Instead, here you can see the students that are currently working with me. all except pilar, that instead was involved in one of the gut bacteria. And Anna and Imma stay with me since a lot of time, and they contribute on all the work on giant viruses. And these are my PhD with Antonio Lemo now, postdoc, and Alice and Antonio, the last one, instead, they are undergraduated. I thank also a lot of all the collaboration I have, and the great one is with Professor Jane Manhattan for the chloroviruses, the very last part of our talk. And this is a picture of my town. And now you can understand why I was so eager to come to New Zealand, because I feel at home. I like your country a lot, because you have the sea like in my country and a wonderful landscape that I really adore. And so once more, thank you for giving me the opportunity to visit your lovely country and to tell you some stories about carbohydrates in this occasion. Thank you.
I want to borrow this. Thank you, Christina. That was awesome. Giant sugar-covered viruses, that scares me. But what I'd like to do now is just ask the audience if they have any questions for Christina. Yeah. Anne.
Kia ora. Thank you very much. I have a question actually going back to the interaction between your DC sign and TLR4 and looking at how the beneficial bacteria might be altering that activation. Do you think that it's by preventing that clustering effect? So in fact, they're competing for either the DC sign or TLR4, preventing that and therefore dampening down the signal when you have, say, the beneficial or the E. coli TLR?
It's an interesting question because, when you have, the idea is that the polysaccharide is promoting the clustering, so it's not preventing the association, but it's promoting that. And in this way, it's modulating the response. That's the point between, so that you have TLR and DC sign and the polysaccharide bridging them. But that's the case for vulgatus. So we cannot make it general because in such a scenario, each polysaccharide is a different story, and I only showed you one. Or you think that maybe it's preventive, but in theory they are close to each other, so there is clustering.
Yeah, well, I was just wondering whether or not there's a different affinity for one versus the other, the different types of LPS, and by doing that, changing that clustering. But I can talk to you later.
Okay, that's great.
Lovely.
Even because I hear and I don't hear you very well, so that's fine.
Thanks, Christina. That was it was amazing. So just the first bit, I was trying to think about how to make the question simple, but I'm going to probably struggle. So, so the first bit of the question is just a fact check, for me and for the audience. So, from the first bit of your talk, you beautifully explained that you linked the reason why it's good to have diverse microflora in our gut all the way back to the fine structure of the lipid A core molecule. Was that correct? Yeah, I think so. Hopefully I got that bit right. So, and I know I should know the answer to this, but I truly have forgotten. For the activity that you put up on the board, so it was either inflammatory, it was immune activatory, or I think blocking maybe. Does the binding, can those molecules still bind with the extensive glycosylation state, or do the sugars have to be removed? Because normally in the lab, when we test the lipid A structures, we've removed most of the oligosaccharides. So my question is, do they still bind? That's the first question, with all of the oligosaccharides.
I have to confess that I don't understand the question.
Okay. Where I was going with the question was that in the endogenous state in our stomach, or in our GI tract, the pH is going to change a lot from the stomach as we move through the GI tract. And I was just wondering the impact of the acid sensitivity of the glycosidic linkage that you talked about, with the cleavage, and whether the different fine structure could influence the sensitivity to acid of the glycosidic linkage. Yeah.
First, the story of the pH, which I got it. So, and the pH, yes, is going to decrease, but it's not, are no conditions that are going to cleave the lipid, let's say, from the rest of the molecule. And even if this was going on, the lipid will still be the immunoreactive part, so it will be active against the TLR4, at least for the E. coli lipid. So for E. coli, there is no problem, but in any case, the pH is not such a strong to cleave the molecule in parts, also for the good bacteria, for Bacteroides. If that was the first part of your question, but then the second you have to repeat it once more. So I'm sorry.
I think you've answered the question, so that's great. Thank you.
Any other questions, or can we feed and water our speaker? Okay. If there are no further questions, thank you so much, Christina. That was an awesome talk. It's kind of scary hearing about, how subtle changes in sugar structure on the surface of these organisms can, trigger our immune responses. As someone who suffers from autoimmune diseases, I kind of worry about some of this stuff. So that was really, really cool. Thank you, everybody, for coming along. It's so much appreciated. If you want to be part of our mailing list, there's a QR code on the side that you can, take a picture of and join our mailing list so you can get emails in future about these events. And I just want to thank Christina once more for a fantastic talk.